everyone, and welcome to this latest installment of Breaking Down Barriers, where we're going to be talking about a topic that we've kind of covered in some different sessions, but it's really important to continue to talk about because of how science is focusing and looking at this a little bit more intentionally is the idea and topic of somatic instability or expansion. So I'm really thrilled to have some amazing people join in chatting with us about this. First off, I wanted to welcome Dr. Steve McCarroll. If you wanted to introduce yourself.
Hi, I'm Steve McCarroll. I run a biomedical research lab that's part of Harvard Medical School and the Howard Hughes Medical Institute and the Broad Institute. Um, I spend most of my day mentoring and coaching the scientists in my lab as they try to uncover how the brain works and why it's vulnerable in the specific ways that it is. It's a
Great,
it's a
thanks.
joyful job that involves a lot of teaching and mentoring and on good days, insights and discoveries.
That's amazing. Thanks so much for being here. And like we always like to do, we're including one of our ambassadors because we feel like it's important to have the community perspective. So today we have with us Erik.
Hi, everybody. I'm Erik. I'm currently 23, living at risk in Calgary, which is located in Canada. Um, I've been involved with HDYO for about 3 years now as an ambassador, mostly doing advocacy stuff like this, as well as lots of fundraising and spreading awareness in my local community. But yeah, I'm just excited to get to know a bit more about the science behind the upcoming research around HD.
This is great. Thanks for being here, Erik. And really, with these breaking down barrier conversations that we have, the concept is that there's a lot of things that we hear about. It gets really confusing. And it's important to have these ways of talking about it to make it much more conversational. So, as you are learning more, if you're interested in participating in different science or experiencing different things, you have the ability to really start or enhance that understanding of what's being talked about. Erik, I'm going to start and pick on you. Why did you want to join this conversation when I reached out? Why is this important to you to learn more?
Yeah, I think it's a lot of what you said, like enhancing your understanding. Like somatic instability is a newer term that I heard a couple of years ago at a conference. Um, but I don't really know like how it impacts me, if it's something that I should be worried about or like trying to talk to my doctor about. And like, I don't know, just there's a lot of questions around it. So I just wanted to kind of peel back the curtains from an expert.
Yeah, I think this is great. Um, Dr. McCarroll, we always like to start by getting to know you a little bit more. So you shared a little bit about what you do day to day, but do you mind telling us a little bit about how you got involved in the Huntington's disease community and in research?
Yeah, so my lab had been working for many years to better understand the biology of the brain, using genetics as a tool, using DNA as a tool, and the expression of genes as a tool, the decisions that individual cells make to turn genes on and off as a way of learning about those cells and their function and their health. And about a decade ago, scientists in my lab invented a new way to study how individual cells are using their DNA. So to basically study for thousands of individual cells at the same time, which genes each cell is turning on and turning off. And it's a way of trying to understand the biology of the brain, but by studying each of the cells as individuals rather than just studying kind of the the sum total or average of them. And we made this invention available to all scientists by posting blueprints for it online, and thousands of labs built their own ones. And the scientists in my lab started using it to study the cells that populate the striatum, a part of the brain that's severely affected in Huntington's disease. And we discovered a series of surprising things about them. And then what led us to Huntington's disease actually is there's a foundation called the CHDI Foundation that supports a great deal of Huntington's disease research. And it's actually scientists at CHDI who became aware of our work and reached out to us about getting us involved in Huntington's disease in particular, rather than just kind of in the brain and the striatum in general. And they really brought us into the community. It was a really welcoming community of researchers, and they supported our work and helped us design and pursue a whole new project of several years.
Yeah, that's amazing. It's great to understand that because I think it's important for the community to understand that there's a lot of cross collaboration that happens in science because like yourself in your lab, you weren't necessarily focused on Huntington's disease, but there are lots of benefits that can then be further studied with what you were doing and how it crosses over in HD. And we also see that in research being done of drug repurposing too. If there are certain therapies or drugs that maybe are in another disease area that could have an impact in Huntington's disease, then you may see that being looked at too for Huntington's disease because of some of those correlations. So there is a lot of correlating factors that happen in science. And I think that's really important for the community to know.
Oh, it's so true. We all advance each other's work in hundreds of different ways, actually, and it's important to recognize that. It really, science is a team sport, and the Huntington's disease community has the Huntington's disease scientific community has been particularly welcoming. We were the new kids, and other researchers in the field taught us a lot about things we didn't know, but that were very helpful for us to understand. Especially researchers who had, like Sarah Tabrizi, who had clinical backgrounds and clinical experiences and could really help us connect the basic biology to those experiences. But really just thought that whole scientific community was really welcoming, and that that helped us do our work better and more quickly. And I hope that what we've discovered also, informs and just kind of helps everyone do the best possible work as quickly as we can.
No, I love that. Thanks so much for sharing that. And for the community out there, sometimes you all hear from CHDI in reference to them facilitating Enroll HD, which is the largest observational studies in Huntington's disease, but they have such a hand in so many different research projects and kind of serve as this incubation unit to be able to fuel research, to be able to find comprehensive treatments and therapies for HD specifically, which is something that other diseases don't have. So it's a really important thing and benefit that HD has.
And that's exactly what they did in our case. They incubated this project from the time it was a baby project, and up until the present day.
Yeah, that's awesome. Well, let's jump into the topic at hand. But first, I wondered, Dr. McCarroll, for those to just kind of level set, can you remind us a little bit about what Huntington's disease is, what it looks like in our genetics, how it's passed on, those kinds of aspects of it?
Yeah, so the genetic cause of Huntington's disease was discovered about 30 years ago by an international team of scientists. It was actually a historic uh, discovery. It was one of the first such things discovered in human genetics, and the excitement about it led to the effort to sequence the whole human genome, actually. So it's really a historic moment for human genetics. The international team that discovered the Huntington's gene found that, like all genes, we all have two copies of the gene, but and each copy or version of the gene is called an allele. And they discovered that that all copies have a CAG repeat. So we all have two copies of the gene, and we all have copies with a repeat of the DNA sequence CAG, CAG, CAG in it. But what they discovered was that if that repeat is longer than about 35 or so CAGs, that essentially everyone with Huntington's had at least one copy that had 35 or more CAGs, and that was basically the cause of the disease. And then that made possible then very much biology research to try to understand why the length of that repeat and how the length of that repeat affects the biology of cells in the brain.
Mhm. Yeah, I think that's really important. Thanks so much for sharing that. Um, as we go through this conversation, if you all are interested in learning more or how we'll try to break down these research terms. But we do have a research terminology that we'll add in the link for here as well. So if this is something that you wanted to learn more about some of these biological factors of Huntington's disease, we have a whole section on that, plus some other terms that we'll talk about as well, which could be a helpful resource. One of the things that that the community hears too is that there can be a correlation with the CAG repeat and the onset of symptoms. Can you talk a little bit about that?
Yeah, so the same international team that discovered the gene discovered that the longer the CAG repeat, the earlier in life symptoms tend to appear. And that's true. It's true in average. It's not like a perfect predictor or anything, but certainly on average, there's a quite strong effect where inheriting a repeat of 40 CAGs tends to involve a much later onset than a repeat of 50 or 60 CAGs. And and because a child will tend to inherit most of the time, not every time, but most of the time will tend to inherit a CAG repeat of the same length that their parent had. It's true that on average, parents with later onset tend to have children with later onset, and the same thing for earlier onset. It's true on average, but it's just a correlation. It's kind of the same way that like tall people tend to have taller kids, right? And yet we all know plenty of people that are way taller than either parent. So it's something that's true on average, but it's not like a precise timer or anything remotely like that.
I always say that statistics are great because it shows that correlation, but people aren't statistics. So the individual person may fit in with this correlation, but you have your own individual experiences, and especially with genetics, that gets really complicated too. So statistics can be really powerful, but each individual person can have different variations with that. So I think that it's good to know both of those things.
Absolutely. And that's where we get to another one of the scientific insights that led up to what we did, which was that there was this work by another international team of scientists, actually included many of the same people from the first team, that then sought out to discover why do some people develop symptoms earlier or later than you would have expected based on the length of their CAG repeat. And what they discovered is that that's substantially shaped by other genes. So, and that they actually found many of those other genes that cause one to have earlier or later onset than you would have expected based on the Huntington gene version that was inherited from one's parents. And because each of us transmits only one of our two gene copies to a parent, a child, even if they inherited the same Huntington gene allele as their parent, doesn't necessarily inherit all their parents' modifiers. They only inherit half of them, and they inherit half of these modifiers from the other parent. And so that's where some of the unpredictability enters the picture in terms of aging.
Yeah.
Yeah.
That's really well said. Thank you for that. Erik, I'm curious from your own perspective and what you've heard from the community, because I know you've attended Congress and camp and you've gotten to hear from a lot of different people globally and locally in Canada. Do people worry that how their parents' CAG may impact them as far as age of onset? Do people think about it? What is your thought?
I think absolutely, like even before I saw that graph that you were talking about with the correlation, age of onset to the CAG repeat, I think it's just something intuitive that people understand. Like the lower your CAG, if you're very low, then you won't develop symptoms at all. But then people with juvenile have very, very high CAG. So I think it's definitely something that people look at. But I think there's still a lot of questions around, like you mentioned, the genetic modifiers or even like lifestyle changes that people can do to try and shift the age of onset or even like slow the progression once they do start developing symptoms.
Yeah. Do you ever think do you ever think I have mannerisms of my parents, so I must be similar with because I know you're at risk. So do you ever worry that because you may favor your father as far as different behaviors or different physical attributes, that that is an indicator of just if you're gene positive or not? We do hear that from people, like, I have blue eyes. My parent who has HD has blue eyes, so I must be a gene carrier. I must have symptoms similar or have that similar kind of makeup.
Yeah, exactly. Like it's definitely, I'd say, a common narrative with people, whether you're like at risk or even um, if you are positive for HD. I think for myself, I definitely catch myself like symptom hunting in that way. Like, um, people say like, oh, you laugh like your dad or like stuff like that. So I think it's definitely there. And I don't know what the science says on stuff like that, but I always just try to kind of temper those thoughts when they come up.
Yeah, it's the most natural thing in the world to think things like that. On some level, we're all kind of armchair geneticists. But that specific thing is not something we need to worry about. So we all inherit one copy of each gene from each parent. We have essentially the same inheritance, the same amount of inheritance from our mom as we do from our dad. And the Huntington gene is just one of those 30,000 genes. So the fact that we see similarities in other ways, whether it's how we laugh or talk or the color of our eyes, um, is not a clue about whether we share the Huntington's gene. We just share half of all our genes with our mom and half with our dad.
Yeah. Well, this is great, and this leads right into the topic at hand with somatic expansion. So first of all, you shared a little bit of a tee up for what somatic expansion is, but can you dive a little bit deeper into what it means, how it impacts, what some of those studies really focused on? Are there certain specific genes that we found it in? Could there be more? Let's talk about it.
Yeah, yeah. This is a really interesting area of science because it's an area where there are things that we thought historically, but recently a lot of new information has been changing how we think about it. So, I think we have tended historically to think of our DNA as stable, that we inherit a set of genes, DNA sequences from one parent and another set from another parent, and that then every cell has an identical copy of that same DNA, and that that's stable throughout our whole life, like, except maybe in cancer cells where we know there's lots of mutations. It's what we thought for a long time, and it's still true for most, I would say almost all of the DNA that we have. But DNA repeats are a little different. And starting about 25 years ago, work by Vanessa Wheeler and Peggy Shelburne and other scientists started to teach us that DNA repeats are different in this way. They are mildly unstable in the sense that within a cell, they may change in length during our lifetimes. And this tends to be by incremental mutations that'll make it get like one CAG shorter or one CAG longer at a time. For reasons we don't know, the lengthening ones seem to happen more frequently than the shortening ones, such that over a long period of time, where there's been multiple events, these repeats on average will tend to grow in length. And Vanessa and Peggy and others showed that this instability is particularly pronounced in the brain for reasons we're actually still today working to understand. And that it's especially so in the brain areas that are most affected by Huntington's disease. So this was an early hint that somatic instability might matter in this disease. And then another hint came recently in that genetic modifier study that we were talking about a few minutes ago, because many of the genes that they found to have these quantitative effects on age at onset that would make people tend to have symptoms a little earlier or later than you would have expected, those are in genes that have roles in that seem to have the ability to regulate the stability of DNA repeats. And so that was another hint that this somatic instability of the DNA repeat might matter to the onset of this this disease.
So in someone who maybe doesn't have one of these genetic modifiers that would make that instability happen much earlier, um, to my understanding, correct me if I'm wrong, there is expansion of the CAG repeat over time. So as someone gets older, they may have a 41 repeat, but then let's say 20 years down the line, that repeat expands, which then as they just get older naturally, but with somatic expansion and these other these other modifiers that may be at play, that just happens at a more rapid pace. Is that fair to say?
Yeah, so it may be useful to also keep this in mind too. So we all have modifiers. The modifiers are actually very common. And so everyone has modifiers. Some are onset hastening modifiers, some are onset delaying modifiers, and there's many of them. So we all have them, and we all have them in different combinations and amounts. It's a bit like the genetics of height in that way. It's not caused by just any one gene in a simple way. And so the kind of net effect of many modifiers will, of just a person's own kind of portfolio of modifiers will tend to make them will tend to lead to their developing symptoms earlier or later than you would have expected. But these and the same modifiers seem to affect the level of instability, the frequency with which these incremental length changes are happening over the course of a person's life. So it's as far as we know, somatic expansion actually happens in everyone, but just at different rates. And having
Yep.
enough of the protective modifiers will just cause it to happen less frequently. And therefore, over the decades, it will lengthen more slowly.
Erik, are you tracking?
Yeah, yeah, no, the biology makes sense. Yeah.
Good. Awesome. So I wanted to dive into a little bit more specific about some of the publications that you had a paper that was published in January. You talked a little bit about some of these things that you were working on. Can you share a little bit about what specifically you all were focusing on within your lab?
Sure, sure. So let's see. So we talked briefly about this technology that our lab had invented a decade or so to study that we called DropSeq at the time, or where it's basically single cell RNA sequencing. So it just lets you study how thousands of individual cells are using their DNA, which genes they're turning on, which genes they're turning off. And that both tells you about the identity of each of those cells, like what kind of cell it is, what kind of neuron it is, um, but also something about its health. So we can tell if a cell is sick, for example, by the genes that it is turning on. It may be turning on genes that are helping it cope with some kind of a stress or insult. So first, just imagine this way of looking at the brain, where instead of kind of looking at it in aggregate, you sample thousands of the individual cells in it, and you ask of each one, well, what kind of cell are you? What are you up to? And on some level, are you healthy or not? And so that technology that we had developed made it possible to sort of study the brain in that way. And then the new capability that we added to that technology for this project was the ability to also, in each of those cells, measure the length of that specific cell's CAG repeat. So we could tell if that cell had expanded its CAG repeat a lot or just a little bit. And so with each individual cell, we could tell, was it somatic expansion, modest, or was it somatic expansion very long? And that let us ask the question, are the cells, at what length of the CAG repeat does the cell start to become sick? And what's that relationship kind of between the sickness of the individual cell and the length of that cell's own CAG repeats? It's really letting us ask that question at the level of the individual cell rather than the kind of at you know at the level of like the whole tissue.
Well, that's really interesting. How do you see that impacting research as far as what therapies are being looked at. We've heard a lot about Huntington lowering, whether that's in the form of an ASO, if that's in the form of gene therapy, if that's in the form of splicing. There's a lot of emphasis in lowering either both or the mutated copy of the Huntington protein. So how do you see this playing into that, knowing that there's this whole other story happening too in some cases?
So before we jump to that, let's actually just talk about what these experiments taught us.
Great.
Because
Perfect.
that then shapes
Yep.
sort of how it might change where we see like new therapeutic possibilities arising. So these experiments taught us something quite surprising, which is that the CAG repeat doesn't even begin to become toxic until it's become extremely long. We estimate about 150 CAGs. This was not the way that we or most people had been thinking about the disease before. I think for a long time, biology thought that that the CAG repeat that causes the disease is just born toxic. It's just that it's only slowly toxic, and it takes decades for that toxicity to build up enough to bring about symptoms. But what these experiments taught us is that actually that DNA repeat may be innocuous in the form that it's inherited and may remain innocuous even after it's expanded quite a bit. And that it's only after it's become extremely long that we see the health of that specific cell that has that really long repeat starting to decline. And then it seems to decline rather quickly. So in many ways, I think the truly surprising thing that these experiments taught us is that for almost all of a cell's life, it may not actually have a toxic gene, just a kind of ticking DNA clock. And that gene may only become toxic in the last few months of that cell's life. And then this is happening at different times in different cells. So different cells kind of become sick and are lost at different times. But for most of a cell's life, it's not a toxic gene, just a ticking DNA clock. This really changed how we think how about the disease arises in our cells.
No, thanks for clarifying that. Yeah, absolutely. Erik, curious about your thoughts on all of this. Is this what you
Um,
expected? Is this new?
I think the fact that it, like, once it only hits that around 150, does it become toxic was really, really surprising to me that, yeah, it's not just like the ticking clock. I'm really curious, like, if there would be any lifestyle factors or other things that we can do as people. Like, I'm 23, so if I do have HD, like, what are the things I could do to try and slow somatic expansion or other stuff like that?
That's such a great question, and I wish we knew the answer to it today. It's quite possible that there are environmental modifiers. We just don't know about them yet. But I think this is quickly becoming an area of focus of biological studies in Huntington's disease. And I think that's going to be the question that you just asked is going to be a really natural question. Are there lifestyle interventions that could slow this down? And of course, the exciting possibility is that of potentially someday being able to modify it with medications as well. But in terms of lifestyle interventions, we may well just discover some at some point. We just don't know about them yet. So I think for now, all we can say is to do what gives you the greatest possible vitality and the most amount of life and health every day.
Yeah. I know we talk a lot about some of those coping mechanisms and meditation, and those aren't necessarily altering the disease progression, but it's helping manage some of those emotional stressors, some of those traumatic stressors that you've had that kind of play into some of those factors, or living a healthy lifestyle. We know that that's just a good mode to have anyway, because yeah, there are just so many unknowns, but we do talk a lot about that. Doesn't change the biology, but it just helps with those everyday kind of aspects of life.
Oh, absolutely. And
Yeah.
science is discovering all the time ways that things like meditation and prayer and
Hmm.
mindfulness have powerful
Hmm.
effects on our physiology. And it may we may well and almost always beneficial effects.
Hmm.
It's essentially always that I know of. So like it may well be that in the future it will reveal to us that this very specific ways in which
Hmm.
this affects positively our health. We just don't know what that picture looks like yet.
Absolutely. Yeah. No, absolutely. Okay, so now back to the question, what does this mean for research? What does this mean for therapy development?
Yeah, so we now realize from these experiments that a brain cell spends more than 95%, maybe as much as 98% of its life with a Huntington gene that isn't actually toxic, just
Mhm.
unstable. And the threat comes not from this kind of creeping, slow biological toxicity, but
Mhm.
just from the likelihood that someday down the road, that ticking DNA clock will get to a problematic point that that DNA repeat will get long enough to harm brain cells. And at different times for different individual cells, because not all cells are expanding at the same rate. So, but for most of our lives, it's not a toxic gene. That at least that's what we think. That's what we think these experiments suggest. It's just more like this ticking DNA clock. So we think
Mhm.
then that there's just enormous opportunity to try to develop treatments that slow or even stop this ticking DNA clock. And
Mhm.
that such treatments could, in principle, prevent or delay the onset of the disease. And
Mhm.
that even once symptoms have started, that they could, in principle, slow or perhaps even stop the progression of the disease. Obviously, it's
Mhm.
going to take a lot of work by a lot of smart, hardworking people to get to that point of having therapies. But I think it helps a lot to know that how much potential therapeutic leverage there is.
Mhm.
That this is potentially just a powerful place at which to try to intercept the
Mhm.
the the, the pathology in maybe in some cases before it starts.
Mhm.
And it's a matter of figuring out how to do it, which is still always a hard problem. But there's some
Mhm.
important insights from the work of other labs, actually, that I think suggests that although it's drug development's always a hard problem, that this may be a quite feasible one. And
Mhm.
one of them is that these are this somatic expansion um, isn't just something that happens to our cells. It is actually something that our cells' own proteins are causing to happen. And the specific, very specific protein, I'm sorry, somatic expansion isn't something that's just happening to our cells. It's something that our cells' own DNA repair enzymes are causing to happen. And that there's very specific enzymes that Vanessa Wheeler and Ricardo Moropinto and other scientists have whose roles they've discovered that cause these incremental expansion events to take place throughout our lives. And the compelling therapeutic idea is that if you reduced the activity levels of those enzymes with a medication, that you could slow the rate at which these incremental repeat expansions happen over the course of a person's life and potentially postpone onset so much that it's not a factor for people. That's the place
Mhm.
we'd all love to get to.
Mhm. Eric, what do you think this means to the community?
I think it's definitely hopeful. Um, I think it's really cool that it kind of offers a new approach to different treatments for HD. I think people in general are always very excited to learn about the research, and I'm excited to see how different conversations, like the next conference, I'm sure a lot of people will be talking about this. Um, Yeah, I mean, definitely hopeful. I think people will have a lot of questions and they'll be wanting to follow it really closely.
Well, what we see too is that there are some new trials or kind of reconfiguration of therapies that are looking at how does it affect some of these known modifiers, in addition to the initial endpoints that they're trying to reach, whether that's Huntington lowering through the different ways or other approaches. Dr. McCarroll, for those people who are considering participating in some of these current or upcoming trials that maybe aren't looking at somatic instability and expansion, do you think they should wait until something else is discovered, or do you think if they, if there's a clinical trial that they're really interested in, and are eligible for, that they should consider joining that trial?
It's a wonderful gift to get involved in research. It allows one's story to inform everyone's understanding and help the field take the best possible, most well-informed next steps on the journey to discovering ever better therapeutics. And every research insight helps everyone focus better on the best, most promising things. And contributing to that process in any way is one of the greatest gifts we can give one another.
And I think too, for a lot of you listening and watching out there, you may not be able to participate in a traditional clinical trial for lots of reasons, whether it's access, whether it's being at risk versus knowing your genetic status, whether it's being pre-symptomatic or what have you. But there are so many ways to get involved in research. And we mentioned Enroll HD. They're really focusing on, in this next iteration, looking at the earlier stages of understanding how the brain is developing earlier before symptoms are known, for the HD community. But plus, there are other observational studies. You may have a clinic nearby that has something. We have our survey series, which is really important because it understands some of the ways that you feel and think and how we can do better and work with our partners. So we can't emphasize enough the importance of sharing your perspective in whatever way that looks like. And there are safe ways of doing it anonymously. There are safe ways of doing it where you don't have to know your genetic status to participate. But research in whatever form is so helpful, but it's also empowering for you, is what we hear as well. Erik, I know you participate in some observational or survey studies. What does participating in research mean to you? What do you feel like once you do that?
I think a lot of it is it feels like you're not running from something, but you kind of turn to face it. And like, I don't have a biology degree. I'm not looking to spend 5 years doing a biology degree to get into research. So it's a way that we can still give back in a way that's really meaningful. I think it's a similar feeling to like if you're involved with any advocacy or fundraising or even just volunteering around your HD community, it's a similar feeling where you just know that your actions are contributing to something bigger. Definitely helps quell a lot of the negative feelings that come up around HD.
Yeah. And I think, there are so many fears, and I address some of them, but there are safe ways to participate. So if it is something that you're interested in learning how you can get involved in any kind of research, reach out to us. We're happy to talk through what some of those options are. We had an observational studies dating game that we did at our Congress, which was pretty nuanced. And I dressed up as the 1970s game show host, and we had different observational studies. Dr. McCarroll's looking at me like, what the heck is that? And Erik, you participated, and just the ability to be able to say, these are some observational studies. This is what's important to me. How can we get matched up with one that meets my needs, the time commitments, my comfortability with sharing information, all of those kinds of things. So we'll also put that link in here too, because it's a good way of understanding some of the things that are out there, understanding what some personal profiles are for community members, and then how to kind of match them up based off of their needs. So we're happy to do that individually as well and talk through options if that's something that you're interested in.
Yeah, if I could jump in on that, I would
Yeah.
say like for anybody that's at risk, you can still participate like I do enroll HD. So I would definitely go check out that link that Jen is going to show
Yeah.
because um, education's important and
Mhm.
it's always good to be involved.
And that
I'd
video
love
is really funny.
oh, I'd love to add one more thing about another way that families can contribute to research and discovery in HD. Uh, so it's the deepest and most heartfelt goal of everyone in this field, every doctor, every scientist, to give everyone the longest and healthiest possible life with the greatest possible amount of vitality. And the focus on that sometimes prevents us from having the following conversation, which is that it's also a gift if at the end of life,
Mhm.
whether one's, and this is true of everybody, whether they're at
Mhm.
risk or not. Um, if at the end of life we donate tissue to research, both our own brain tissue, but also probably a more present thing for people is family members who may pass away before we do. It's like being an organ donor, but just another way that we can uh, contribute to knowledge and understanding. And everyone in my family signed up to do it, but there's not a lot of
Mhm.
awareness of it sometimes, or not as
Mhm.
much as there could be. And it's a powerful way to contribute to the science in this area. Everything that we discovered in this work, every single thing came from brain donations that
Mhm.
people or their family members made at the end of life. And these are scientific gifts that enabled all the work that we did and continued to enable the work that we do now, um, in all kinds of ways.
Mhm.
Everyone is a story, right? And a story that can teach us something. Someone donated to research who it turns out had a HD causing allele where might have expected her to develop symptoms in her 40s, and she lived to her 80s without developing a single symptom. One person can teach us a lot, actually.
Mhm.
And so that's another way that people and their family members, whether affected or unaffected, can advance the cause.
Yeah, absolutely. I think, and we want to just share appreciation of everyone who participates in research, because we know that it takes a lot of effort to do it, and it is a very personal decision. But we're so appreciative of everybody who does donate tissue samples or organs or participates in sharing your perspective, or in observational studies or clinical trials. It's just tremendous the amount of sacrifice and giving that this community continues to do on behalf of others, and we're just very grateful for that.
Yeah, yeah. And I also would just love to mention my colleague, Sabina Beretta, who is my partner in the whole project from planning it from its first days. And uh, she's a terrific neuroscientist, but also, in addition, had played an amazing role over the years um, in shepherding the collection of these brain tissue resources that made the whole research project possible, and interacting with families and things, and so she's an amazing partner in every aspect of this work, actually.
Yeah, that's great. Well, as we wrap up the conversation, Dr. McCarroll, how can people stay tuned to what you all are working on, and just the topic of somatic instability and expansion in general?
Well, it's a community with lots of great uh, conversation about research and emerging
Mhm.
research, and I really love HD Buzz
Mhm.
and other resources like that. Um, so it's a very lively community of families.
Mhm.
There is something unique about this
Mhm.
relative to a lot of diseases, and the extent to which that there are a lot of things that just come take people completely by surprise, right? and like, let's say cancer, right?
Mhm.
This often isn't like that, but one of the things that has come out of that is these wonderful supportive communities that are building, contributing to one another's resilience and strength even ahead of time. And
Mhm.
so that's a really amazing thing.
That's great. Well, any closing thoughts for either of you? Eric, I'll start with you.
Um, Nothing super big. I definitely have a few questions I'm gonna go try and research myself about it. But I definitely excited to see where the research goes, very hopeful for it. I think it's always nice to have a pulse on where the research is progressing, and
Mhm.
especially when there's I guess, new breakthroughs like this, it's definitely a beacon of hope for a lot of people.
Mhm. Dr. McCarroll, any closing thoughts?
So yeah, it's been such a joy to get to know both this community of patients and families, and also of scientists and researchers who had have been working in this space for a while. And everyone's taught us so much, and we're so glad that we've been able to contribute. And we're only getting more involved. So we just expect to still um, continue this journey, and hope we can keep contributing to this mission.
No, I'm so appreciative of both of you. And as we always do, we want to encourage you that when you're at events, please stop the scientists and researchers and chat with them, because it's so great to have those one-on-one connections. You can share your perspective. You can ask questions directly to them about what your concerns might be or your experiences. But that just continues to bridge the gap of having that true community support. One of my favorite things about our Congress is how engaged young people are and asking those questions.
Yeah.
And I would just say keep asking questions, because that's the best way to challenge researchers, but also for you to continue your learning journey.
Can I just chime in on that?
Yeah.
Yes. Please keep asking questions and do approach scientists. We're not too busy. We're not, it's like this is what it's about for people. And it is just it it these interactions with patients and families at conferences just infuse this work with vitality and drive. And please approach us, ask questions, start conversations. It's very welcome, and in fact, it's what it's all about.
That's great. Well, thank you both so much. We're so appreciative of your time. Thanks for all of you for watching. As always, if you have any questions or need any support, we're always an email away. Thank you all so much and take care.
Thank you.
Hi, so we already said goodbye. However, it was really interesting because we're back. Afterwards, Eric asked some really key questions, and we had already stopped recording, and it was too late to hit record again and still capture the same conversation. But it was really exciting questions that Eric had. So we wanted to give him an opportunity to share what questions he asked and then some of the feedback we got from Dr. McCarroll. So, Eric, take it away.
Yeah, still getting used to this whole video format. I should have asked those when we were recording before, but I was really curious because I don't have a big background in somatic expansion, so I was
Mhm.
curious if that happened just in the brain or the blood. Kind of if if I test negative when I'm 20 something, would I end up developing like to be positive later? And his response was mostly that they don't know exactly why it is this way, but somatic expansion typically happens a lot more in brain cells than in
Mhm.
blood cells. So you might see a tiny increase in blood cells over somebody's entire life, but there's never really been evidence to say like somebody wasn't like
Mhm.
gene positive when they were 20, but then they were when they were 50 or 60. Um, it's mostly that the instability with higher CAGs in the brain causes more instability and just like kind of the domino effect in there. Jenna, did I?
Yeah, and I think his point was is that that's why Huntington's disease is known as a brain disease and not a blood disease, and that was an
Right.
interesting way of putting that. And he did say that now that they have found some of these, or found the expansion that happens in the brain, that they're looking in at other tissue too, because um, people don't get brain samples taken out while people are alive. So that's why the brain donation was really important. But now that they know that it exists, there are scientists who are looking at other tissue samples, like in muscles or skin or in liver biopsies or blood samples, because it would be much more convenient and more realistic if we could do measurements of expansion actually in a much more feasible way of getting that biosample out, which we thought was really interesting.
Yeah, and that kind of led into my next question, which was how do
Mhm.
they get the samples from the brain? Like I'm
Mhm.
assuming they don't just like stick a needle through a skull.
Yeah.
And that's where like at the end of the call, he was talking about the importance of being like a brain donor or organ donors and stuff,
Mhm.
because you're not like legally allowed to sample somebody's brain while they're alive, which
Mhm.
makes sense.
Yeah.
Um, so a lot of the information that they're getting is from people who had donated their bodies to science.
And I think that that's a really important piece because a lot of research does happen. I mean, research happens in a lab. And then when it gets to a clinical trial phase of it, they've taken the research that's happened in the lab and they've figured out how to apply it into a live person or an animal or whatever stage that they're in. And that's part of the reason of it is that they understand what's happening now, but now it's like, what does this mean? And how can this translate into understanding how it can impact people and into the therapy kind of arena that we have? And that's where it's kind of like where the rubber meets the road. So now that they found this one piece of it, now what does that mean more not mechanically, but more like influential with how people's lives are interacted with what the science is saying? Instrumental, I don't know, one of those words.
Yeah. Um, I think one of the later questions I asked was like if we covered everything he wanted to talk
Mhm.
about, if there's anything kind of in the community that or anything that he wanted the community to know more. Um, and then Jenna, my memory's a little foggy on it, so feel free to jump in, but he was talking about how like how do people perceive um, HD kind of leading up to this? Like, is it like a slow toxin that releases, and then people who like have the gene just kind of live with this like underlying like worry or like constant like thinking about if their brain's gonna like I don't know how.
He was thinking like, do people think it's like a slow release
Yeah,
toxin
yeah,
over the course of time? And what he shared was more that that the brain cells do get damaged, but it's more of a the cells individually, the DNA kind of like flips that switch of now the the decay happens, or now that the symptoms are really going to start, or like the HD somatic expansion happens like more quickly within that. So there's a lot of preservation that happens in the cells until that switch happens. And how does the community view about? Is it more of a long-term kind of toxic slow release over a course of time or flipping? And I think both Erik and I said that a lot of people, it's very specific to where people are at in their journey, because there are so many day-to-day impacts of HD that if someone who's actively caregiving but is either at risk or gene positive, they maybe are looking at how do the day-to-day impacts affect me, or am I like when am I going to start having more of those symptoms that are going to maybe make me have to rethink future planning or work, or some of those more tangible things that HD impacts versus kind of some of the biological factors of timing. I don't know, Erik, you chimed in too about your kind of your perspective of how you've thought about it as well.
yeah. I said I kind of picture it in the same way of like cancer in a way, like you're 50% you have the gene or not. And then in my mind, I'm like, well, if I have the gene, then there's other things that I can do to still delay the onset. It's not like
Mhm.
I have it and then that's like a life sentence, but I can still be like active, manage my stress,
Mhm.
reduce like environmental toxins and things like that that would just contribute to like an overall stress on my body that would
Mhm.
eventually like lead to symptoms developing sooner.
Mhm.
Yeah.
Yeah. So I guess we're curious to hear what you say. So maybe you guys can comment below on what, how do you think of how the disease progresses in your vein? A lot of the things that we hear are it's more about the impacts of the disease progression, less about necessarily um, how the brain is developing over that time. But again, how is being able to work as long as possible, or having cognitive abilities, or what have you. So yeah, interested to hear from you all.
Yeah. I think that was kind of the last part that he talked
Mhm.
about a lot was like as a researcher, he doesn't have the perspective like of a lived
Mhm.
family going through the stages of HD. So he was really curious, and like he said, he really likes to talk to families whenever he's at
Mhm.
these meetings and trying to understand their perspective because it kind of helps them focus more on like where they want to direct research, how they can be
Mhm.
supporting the communities better. So kind of like what we said at the beginning of the first part,
Mhm.
like make sure you're engaging with these people. They care a lot.
Mhm.
I've never met a researcher that seems like they don't have the time for me, which is
Mhm.
really, really crazy considering how academia usually goes. People are usually just kind of up in their ivory towers. So yeah, it was really nice to he spent pretty much half an hour just answering random questions from me, a person he's never really met before. So
Yeah. Yeah, it was a great conversation. And I would say perceived towers. I think that a lot of academics are happy, but someone has to make them the first move. And I think most are really eager to chat with the community, which is really exciting.
yeah, that's
And how we
definitely
learn. Yeah.
good point.
Yeah. I think you've heard me say this before, Erik, and I've said it before. I think part of it's just nerves. Like, am I going to sound stupid? I mean, here's this person who understands how the body works in ways that maybe I don't, but you are each the experts of your own journey. And so you are the experts in Huntington's disease with how it impacts you. They might be looking at the biological aspect of it and things like that, but you are the experts. But if ever I feel like this might be a dumb question, I will always say, like, dumb question amnesty. I'm just curious though. So like, but more often than not, it's not a dumb question. And if even if you're in a group environment, asking those questions is important because I can guarantee you that there is someone else in that room who has that exact same question. So please don't ever feel ashamed or afraid, or that if you're asking the question that you don't get it, because it's more about your learning process, and that will always be respected. Well, this is awesome. Thanks, Erik, for asking those great questions, and yeah, we'll keep you updated. So thanks again for joining.
Thanks, everyone.